So good morning, everybody. Thank you for the introduction, and thank you for inviting me to this very interesting conference. I'm not a computer scientist, and the closest I ever got to a supercomputer was, as you just heard, I was at CERN in the 80s, at the end of the 80s. It was the first supercomputer of Cray that ever arrived in Europe, and I used it to implement the code to track electrons through matter, the EGS code, which some of you may know. That was one of the very first applications, and so I always found this very exciting. As we also heard, I then got more interested in the electronic data processing and computer architectures, in particular also the basic question on what information really is and how is it processed, in particular in biological circuits. Now, I tried to link to what you people talk about here, which is weak and strong scaling, which of course are the key aspects of building large-scale machines made of many processors. And I will argue today that weak scaling is a huge success, of course, for brain simulation. But in order to make the next step, it will be essential to make progress in strong scaling, which I think is a bit harder. The object we talk about is the brain, which looks kind of frightening if you look at it. But if you look to the numbers, it's actually not too impressive compared to what you people do here, for example. I mean, there are 10 to the 11 nodes that we call neurons. And you just yesterday saw the memory card, which carries a terabyte of data or something like that, which is 10 to the 12. So you see, in terms of number of words or nodes, we are basically there. I mean, I'm not saying we can store a brain on a memory card, but we can store a brain on memory systems that are available. The question is, what are we doing with it? So there are other numbers like 10 to the 15 connections mediated through synapses. That's maybe a bit more frightening. But even this, as we will see, can be implemented on computers, probably already today. The real big difference is what's there in the right bottom corner. The connections are dynamic. It means they are not constant. And they have different ranges. They are long range and short range. And they are mediated by a very strange data protocol, which are spikes or action potentials. And that is really what causes the problem, in particular the fact that those connections change with time by constant interaction with the environment. Now, why would computer scientists or people that are interested in data get involved with the brain at all? Well, there are two questions and two answers. And the first one is the obvious one. I mean, why are you using computers for materials? Why are you using computers for the universe? Well, it's to understand how this complex system works and how it is structured. And so understanding the structure and the function of the brain, that's clearly the number one application of supercomputers in neuroscience. So neuroscience, the advanced neuroscience, is the number one application. But then there is a very interesting aspect of what you probably call co-design. The brain itself, of course, is an information processing device. And so by means of understanding it, we will also probably discover interesting architectures that are maybe worthwhile to be transferred back to the computing systems that we use, or even to develop fundamentally different computing systems. So using computational principles of the brain that we may discover during those studies with computers may be used to produce new computing architectures that are generic in nature, so that are no longer only built to produce data or produce information about how the brain works, but to process data in a more generic way, for example, data from business or from other science areas. So like most complex systems, the brain is a very, very structured thing. And so there are scales of spatial extension. And those scales are there in most other complex systems, but they are very, very clearly visible, even under a microscope in the brain. So if you look under a microscope, or instruments somewhat better than a microscope, you see, of course, that the basis is molecules, and below that, atoms and whatever. But basically, it's a molecular device. And then those molecules, they make synapses. Synapses are really almost molecular devices. They are chemical devices. They are very much based on molecular physics or chemistry. And they are the ones, for example, where the learning and the self-organization of the system takes place. And the synapses are very well-defined objects. You can see them under a microscope. If you go up, of course, there are the neurons and other cells like the glial cells on the micrometer scale. Also there, they are physically there. You can see them. And it's very obvious that if you build computer models, that you build those around the scales that are visible under a microscope, that you see in nature. The next higher level are subnetworks like cortical columns, cortical areas, and then, of course, the full brain. So it bridges scales in space from nanometers to, I don't know, 10 centimeters or something like that. Now, having those scales available for your modeling, the question is, how are you doing? How you model a neuron, for example? Again, I think there are two basic approaches. One is to treat the physics of the neuron, the way it communicates, the way it sends information to neighboring cells, as it's called here, an emergent phenomenon. That means you go back to the physics, you try to understand the biophysics of the cell, and you start from the principles underlying the generation of electrical signals, for example, the transport of ions through ion channels, the fact there is an insulating layer, there is a voltage between the inside and the outside of the cell. So that's implementing biophysics, and then see how those cells behave electrically, and they use this as a basis to build your networks. That is an approach that's taken, for example, by the Blue Brain project of Henry Markram at the EPFL in Switzerland. The other one is to treat the behavior of cells, the electrical behavior, here represented by a so-called spike train, as a target function. You say, well, I observe this, and I don't really care what the biophysics, the underlying biophysics is, but I just try to make a model that represents those electrical properties, like firing patterns, for example, and things like that. And that's an approach which is computationally very, very efficient, because you can sort of abstract away all the underlying physics. Of course, it also has disadvantages that you don't have the underlying physics, so you should not expect any insights on the cellular level, but more on the network level. That is an approach that has been taken, for example, by the NEST group, mostly located in Jülich, which, and so I guess there are some people here, or many people of you here, know this kind of approach. And I will talk a little bit about these things and tell you how far we get with these two approaches, and also what the limitations are. Showing you another picture of scales here, which is maybe more beautiful, going from basically from molecules to the full brain. Now, there are tools available, of course, computing tools, and most of them that are used by computational neuroscience today are what we call neuron-based. So there is the neuron doctrine, which says it's the neurons that are the atoms of neural networks, okay? And then, of course, there are the connections and the synapses, and we start at that level. Sorry. Now, of course, there are other ways to model neural networks. One is to go even deeper on the microscopic scale, to really go into information transport in cells by reaction diffusion equations, or if you are very ambitious, you can try to go to the molecular level. You can also say, well, maybe neurons are not the units that really are responsible for information processing. There are higher levels, like, for example, a whole neuron population, mean field approaches, maps, or just Bayesian inference, which you can implement in some kind of code. Now, it's interesting that for all those tools, of course, of all those approaches, there are tools available. I already mentioned NEST for so-called point neurons, where you do not look into the biophysics of the cell. The other one is Neuron, where you actually do look into the structure of the neuron. There is STEPS for the reaction diffusion approach, and GROMACS for the molecular approach. It's interesting to see that, of course, depending on the tool you choose, the memory you need to describe a neuron is vastly different. There's a huge difference. You can describe a point neuron, which just has a couple of parameters by a fraction of a megabyte, probably much less than it's said here. A structured neuron, you need about a megabyte to describe the cell with a decent number of compartments and neurons channels. If you go down to the molecular level, the number gets horrendous, like 100 terabytes or something like that for a single cell. Okay, so if you want to build networks, of course, you have to decide which is the level of this the detail that you really want to implement. Of course, there is the approach of multi-scale simulation, where part of the brain is simulated, say, on the mean field level, and then maybe you have a single cell that is simulated in great detail. But what people do mostly today is to stick to one level of description for a cell, and one which is often chosen, for example, by the group of Henry Markram, is the level where a neuron is a megabyte, which means a neuron is a cell described with some cellular detail, individual ion channels, compartments, propagation of electrical signals along the dendrites and axons. That is a model, as I said, which requires a megabyte. And it's very easy to see that to describe the brain by that kind of approach, you need 100 petabytes, which is kind of frightening, but not totally out of the question. What people do today, of course, is to simulate networks on this level. A single cell is no problem. Even larger structures like so-called cortical columns, which typically represent 10,000 such cells, can be easily simulated on a couple of 100 teraflop machines. And we have reached 10 petaflops machines, as we have seen yesterday. And so today people are ready to simulate cellular circuits on the mesocircuit, that is brain area, or even small rodent brain level. That's what people do today. And what they learn from it, I will tell you later. But these simulations are available. They do it, all right? Now, it's extremely easy to just draw a straight line and say, well, definitely to go to the full brain, we know how many cells there are, 10 to the 10, 10 billion. All we have to do is to get an exaflop machine. And this is one of the arguments why we say, well, we need 100 petabytes of memory, an exaflop machine, and we can simulate the human brain on the cellular level, okay? That's called sort of the rationale behind the idea going to the exascale machine. And I will tell you that there is a problem with this approach. I'm just introducing you to the theme at this very moment. Coming to the concept of scaling, of course, that has been discussed a lot yesterday. Of course, there are two extreme approaches. One is you can go to weak scaling, which basically means size up. And of course, the brain structure basically screams for weak scaling, okay? I mean, you have a cell or a population of cells, which is a fixed size problem. And if you just want to make your network bigger, reaching larger brains, you just exploit weak scaling, and you make the system bigger and bigger and bigger, and you hope that the compute time stays constant. And I will show you some results on that. The other approach, of course, is to chop the problem into pieces and to divide by the number of nodes that you have. And then you get a speed up in principle, which is much harder. Of course, you can, to some extent, chop the problem into smaller pieces. Once you reach the neuron level, of course, it's no longer really possible. So strong scaling is very hard. And it's really, as we will see later, it's the limitation on the simulation speed for neural networks. And I will argue that improvements on strong scaling would be very, very important. And I will show you one proposal. So basically what people do when they run neural simulations on supercomputers is they work in this space here, which is a metric of number of neurons that you simulate versus the simulation speed, the time to solution. And you see the real brain up there in the middle, and that is currently not reachable. And the reason is that it has 10 to the 11 cells. That's one thing. And also, as we will see later, that, of course, there is no way we can do this in biological real time. So the horizontal axis is speed with respect to real time. 1-to-1 means just like our own brain. So your own brain sits at the crossing point of these two red dotted lines. So what people do in supercomputing, of course, as I said, is to exploit weak scaling as much as possible. We'll see how well this is being done. This is an example for a 10 petaflop machine that's in use right now, for example, by the group of Markram, using very detailed cellular models, the 1 megabyte per cell approach, which I have just introduced. And of course, to a certain extent, as long as you have enough processors, the simulation time will be slower than real time, as we will see, but it's independent of the problem, basically independent of the problem size. You will see it's not really true, but if there is perfect weak scaling, that would be the line, the horizontal line. And of course, how far you go out in simulation speed really is then the limit on strong scaling. At some point, you run out of processors. Of course, what you can then do is to resort into serial processing. You can use memory to store intermediate results and then make the network even bigger at the cost of making the simulation slower and slower and slower. It starts to be really slow at some point, and at some point you run out of memory, and that's the end of the game for that particular computing and that particular neuron model, which currently is around, as I said, the mouse brain or something like that. So this is the current picture, of course, in the Human Brain Project with the exascale machine. As I said, it will be possible to extend this plot up to the human brain. Okay, so there will be a simulation, if people want it, on the cellular level of the human brain, for whatever it's worth. And I'm already warning you, it may be not worth too much. I will comment on that, but it could be done technically, all right? I show you the example, which probably you have all seen. This is a simulation run by Henry's group in Lausanne, and you see all these flashing lights. What is shown there, this is actually a network as part of a so-called cortical column, and the color code shows the voltage between the inside and the outside of the cell. All the cells in our body have an electrical voltage between the inside of the outside. Even if you take a skin cell, it's about 60 millivolts, 60, minus 60 millivolts. And special thing about brain cells, of course, is that you can send currents in through the dendrites, postsynaptic potentials, and they will change that voltage. And if the voltage rises, the cell reacts in an extremely nonlinear way, even depolarizing, producing a pulse of about a couple of milliseconds to milliseconds until returning to the resting potential. And it's that process in the network that's being simulated here. Is it interesting? Yes, it is interesting. You can, because this is a biophysical simulation, you can now change biophysical parameters. You can change parameters that you understand, like, for example, behavior of ion channels, arrangement of ion channels, distance of synapses to the cell core, and things like that. And you can see how the firing pattern changes, and you can compare that to real biological data. So in order to tune biophysical parameters, for example, these simulations are extremely useful. There is another approach going to simple cells, and this is work being done by Markus Diesmann, who works actually with the K computer in Japan, which we have seen yesterday. And this is sort of a weak scaling exercise. How well does it work? And what Markus does is he puts processors, 20,000 neurons on a single processor. So there is this processor to cell ratio of 1 to 20,000. You see the network size plotted as a function of the number of compute nodes, and weak scaling works perfectly well. Well, not quite, okay? So what you see, the red dots at the bottom, they tell you the runtime, and that runtime should, in principle, in the case of weak scaling, be independent of the problem size, but you see it's not, and unfortunately it's even a log scale. But it's not so bad, actually. It's kind of flat, and the interesting thing, of course, at some point we have to look at the numbers, the absolute numbers there. And I already tell you that there is something like 10 to the 3 there. That's the time to run one experiment with respect to biological real time. Actually, the simulation speed is about 1,500 to 1, and we will come back to that and discuss this a little bit. Now, seeing this, these are simple cells, actually. It's a very simple network. It may be not very realistic, but it's an interesting exercise in computer science. The K computer uses about 10 or 12 megawatts, and so if you do the calculation, it's very simple. You have 1 billion very simple neurons, 60,000 processors, that's 1% of the brain in quotation marks, because it's not really the brain. It's 13 megawatts. Our brain is, 20 watts. There's a slowdown factor of a factor 1,500. So you can calculate what you pay for the simulation. Because what you pay, you all know that, is not power. What you pay is energy. Energy is power by time. And so there is a factor 10 billion in efficiency, if you want to call that efficiency, between the computer and the biological system. Energy, of course, is always good if you normalize it to a computational task. And what I do here is I normalize it to the energy used for synaptic transmission. Okay, that's why you can also normalize it to action potentials or to a certain number of firings or whatever. I decided to normalize it to synaptic transmission, which is relatively well understood in biology. The energy you use is about 10 femtojoule, 10 to the minus 14 joule. Markus Diesmann on his simulation uses about 0.1 millijoule. That's the factor 10 billion. Henry, with his more complex model, has to pay another factor 10,000. So between Henry and our brain, energy-wise, is a factor of 10 to the 14, which is absolutely tremendous. And of course, the immediate question is, how can that be? And the explanation is actually relatively simple. Do we understand how much neurocomputation costs? Yes, it's actually very easy to calculate. You can do that either top to bottom by using the total brain, and which is 20 watts, which is a good assumption, and divided by the activity, and you end up with 10 to the minus 10 for an action potential, and that's always joule, and 10 to the minus 14 for the synaptic transmission. You can do it also bottom up, which is probably more interesting. Where does the energy come from in the brain? It's hydrolysis of ATP molecules, and we know exactly how much energy we get from an ATP molecule, it's not surprising that it is an electron volt. That's the scale of energy on molecular physics. And so you multiply that with the brain activity, and we know how much you pay for an action potentials and a synaptic transmission in terms of ATP molecules. It's interesting. It's about 10 to the 5 or 10 to the 9 for both of them. And then you end up with the same number. So we basically understand that these numbers make sense in biology. Okay? If you go to electronics, there are things like transistors. Okay? And in a very naive way, we can say computing means charging the gates of transistors, which is wrong, of course. We not only charge the gate of transistors, there are other sources of wasting energy, or sinks of wasting energy. There are leakage currents. There are the connections between the transistors, which are much worse than the transistor themselves. But if you just look to a single transistor, it's an easy calculation. Everybody knows the energy stored in a capacitor, and that's what a transistor is 1/2 the capacitance times the voltage squared. You plug in some numbers, which are old, very old numbers now, 100 nanometer type, at 2 volts, and that means the switching of a transistor, just a single transistor, what you pay is 0.5 femtojoule, which is synapse-like, all right? So a synaptic transmission, as I said, is 10 femtojoule, so you can switch 20 CMOS transistors, and you need the same energy as making a synaptic transmission. So electronics is incredibly efficient. It's as good as biology. Electronics is wonderful. It's just the way we use electronics for brain simulation that is horribly inefficient, because what we do is these things that happen in our brain, we do it by solving differential equations. And we want to do that with a certain precision, and that's just very, very expensive in the von Neumann architecture. And it's not the devices, it's the architecture and the computational model. I think that's very much understood. Now you may say, well, it doesn't really matter, because understanding the brain is one of the key questions of nature. We spend a lot of energy running accelerators at CERN. We send spaceships to Mars, which is very expensive. We should pay megawatts of power for years to understand the brain. It's a basic fundamental question. And I will tell you that even if you're willing to do that, it will not be enough. It is a fundamental thing. And that's the following. Neuroscience has developed wonderful tools. This is the state of neuroscience in terms of tools. This was the state in 1988. This is today, 2014. And these colorful areas tell you the methods. There is MRI imaging, there is electron microscopy, there is patch clamp, field potentials, optogenetics, EEG and MEG. And they cover a space. And what is that space? It's the space of the two scales that are relevant in the brain. It's the spatial scale, which in this particular plot from synapses to the brain covers 7 orders of magnitude. We discussed that. And you see the neuroscience tools cover them as well. So if you simulate, you can compare to the biology, which is great, okay? But there is another scale, and that is forgotten almost all the time. And that's this scale here. It's time. It goes from sub-milliseconds, deep sub-milliseconds, to months or years. That's at least 11 orders of magnitude, which is many more orders of magnitude than the spatial scale. Time is totally forgotten in simulations today. And if you look to the simulations that are being run, like the one of Henry Markram and Marcus Diesmann, they typically run biological timescales of milliseconds, 100 milliseconds. So they sit in the outer little left corner there. And that's because, as we will see, it is fundamentally almost impossible to bridge these large region of brain of timescales. Why would we want to do this, actually? Well, why do you want to look for all these different timescales? Well, why does our brain function? We often think the brain is a structure that's being recorded by microscopes. You can reconstruct it on a computer, then you switch it on, and it will work as a brain. And of course, that's complete bullshit. Okay? The brain only works because it has organized itself. It has interacted with data, and it went through a learning and development process. And that is a slow thing. It's very fast to take a decision. I can recognize you in 100 milliseconds, but it took me 20 years of my life to learn to do this, to learn what faces are, what people are, and so on. This is a study done by Goktai and others. It's the development of many, many brains. MRI study comparing gray and white matter, and what you see here is that gray matter disappears, white matter actually shows up, and that means that connections are developed over a time frame of 15 years. during the lifetime of the human. That's the developmental phase where we learn and where we build an internal model of our world. I'm not saying you have to simulate 15 years, but what I'm saying is that learning computationally is much more costly than the actual application. And learning is the key for application. Networks without functions are pretty useless. That's what I claim. Going back to the computer here, energy is power per time, slowdown factor of 1,500. If you want to simulate a day of biology, you would have to wait 4 years, which is the duration of a PhD thesis. So it's very, very difficult to access those timescales. Why does it take so long? Well, because you have to do both timescales at the same time. You have to simulate synaptic plasticity, for example, on the millisecond or sub-millisecond level, and then the slow developmental processes. And that is a huge challenge which is totally underrated at the moment. Coming back to this plot, which you have seen before, there are things that go beyond just putting the brain on a supercomputer and simulate it for 100 milliseconds. There are many more things. For example, some people want to be more detailed. They want to say, well, we need glial cells, we need reaction diffusion dynamics, we need molecular dynamics. If you want to do that molecular dynamics full brain, you need, what is this, 1 billion exaflop machines, which we don't even have to discuss, of course. Maybe there are other approaches like multi-scale that can help you. But I'm more interested in the bottom 3 things, plasticity, learning, and development, which means beyond exaflop, you need another factor of at least 10, maybe up to 1,000. And that's just not available. I mean, there will be no 1,000 exaflop machines anytime soon. And what we need, that's my claim, is for subcellular detail and maybe more interesting, plasticity that require advances in strong scaling. It means just fundamentally new computing approaches. And really the most important, how should I say, take-home message here is that the only way, I think, to ever make use of artificial neural networks, both for advancing neuroscience and for applying them to things like machine learning, which may be the more interesting thing at the long term, is to give them function. And function, you can give function through theory, through theoretical thinking, implementing and see whether it works. That's one thing. But at the end, it will be key to give function through learning. That means through interaction with the data. And that function through learning concerns the connectivity of the network, the structure of the network, active rewiring on the fly, synaptic weights, and also neuronal plasticity. And that, for me, is a big challenge. Before I go into implementations and neuromorphic computing, let me just, in passing, tell you 2 more things which are interesting from the computing aspect. We all age. I mean, there is not only learning and development, but at least after the age of 20, and you all see where you are on this curve here, and the data is very bad, as you can see. But still, this is a loss of cortical volume in cubic centimeters in the lateral prefrontal cortex, which is responsible for things like planning, cognitive behavior, decision-making, things like that. So it's really a key. This is normal aging, no brain diseases. And you lose a substantial fraction. I mean, if you lose that fraction of transistors on a microprocessor, definitely it would never work. So the robustness of these systems is quite interesting, and we all fundamentally understand why that is. It's the fact that information is distributed in the system, and it's a massively parallel structure, so if you remove part of it, the system still works. It has what we call graceful degradation. And that is a very interesting feature also for artificial computing systems, because it's increasingly difficult to make all transistors work. So to be able to work with non-functioning transistors on the level of 10-20% would be extremely important for the use, for example, of very deep submicron or nanoelectronic devices. There is another thing which is interesting. This is what we may have for dinner this night. It's a lobster, and the lobster also has a nervous system. It's a very simple one. For example, it pushes food through the body. It's a very simple. It's a couple of 10 neurons or so. This is work by IBM Almaden. I love this work because what she did computer simulation. That's a simple simulation. It's just a couple of 10 neurons, very detailed neurons with these, I think it's 17 parameters, which are conductances and reversal potentials of the neuron. And she tries to replicate certain dynamical features like bursts and things like that, which make the food move. And what she did is she ran 2 million simulations with random selection of parameters. She just randomly selected parameters. And out of those 2,400,000 worked. She called them degenerate solutions. That means there's a huge space of parameters where things work. And the variability of the parameters is indicated by those dots here. So you can you can find very, very many different solutions for the same problem, which is also very, very interesting computationally. I have no time to discuss this extensively today, but it's a very important research field of neuromorphic computing. Is the robustness, the resilience against non-functioning elements, and these things which have to do with homeostasis of cells, which both have a huge relevance for computing, I think. But let's go in a more general way now to neuromorphic computing. What does it mean? Putting computers on brains on computers. That's what people often say. That's not the case, of course. We will not be able to do that anytime soon, probably never. But what we can do today is to implement some aspects. What are aspects? I will explain that some aspects of structure and function of biological circuits as analog or digital. I don't care. Whatever you have as a computational model, but I call them images on electronic substrates. Aspects of structure and function of biological circuits as analog or digital images on electronic substrates. What do I mean by structure? Well, the fact that there are cells. That's certainly the basis for current approaches to neuromorphic computing. There are cell cores, the somas, and there's the network, the arms that reach out into the network, the axons and the dendrites, which are active elements themselves. And then there are the connection points, which are chemical processes, but can be modeled by differential equations and then put into electronic circuits or code. Function means local processing, communication by spikes. This is what essentially all neuromorphic systems do at the moment. It may be wrong. It can be discussed. I will discuss it a little bit. But this is what everybody does at the moment. They all say these cells communicate by these action potentials. And then, most importantly, the dynamics, which means the plastic processes, the learning and the developmental things, the rewiring, for example. So this is a very loose definition of neuromorphic computing. And within this loose definition, there are, at the moment, three what I would call really large-scale neuromorphic computing approaches, if you look globally. And they are ordered here in terms of increasing degree of exotics. Okay? What you can do, of course, is to exploit a weak scaling, as I mentioned before, having a fixed problem size and just buying many microprocessors and overcome the problem of weak scaling, which is the communication, by having a really optimized communication protocol so that you can really keep the line flat. This is what the group of Steve Ferber does in the Spinnaker project. I call this soft binary code. So you have ARM processors that are programmed by your favorite programming tools. It's easy to use, and it's commodity processors. There are many of them, and I will discuss this a little bit more. The next one is to leave microprocessors, to say we will not execute code, not even locally on microprocessors, but we build custom circuits. And IBM had a very visible approach, which is now also commercially exploited, which is the True North project. I should have said that here, but it's the True North project. I call it IBM Almaden. It's hard binary code. It means you don't use microprocessors, but you implement neuronal models, like a cell integrated fire neuron, for example, but just wiring up transistors that do nothing else but just modeling that firing neurons. You cannot use it to solve a differential equation. It can only make a neuron fire. That's custom fully digital, IBM True North. Then there is a more exotic and extreme approach, which is what we call a physical model, where you say, now we give up numbers at all. Okay? We say, rather than moving ions through cell membranes, which we do in our brain, we move electrons through transistors. But otherwise, physics-wise, it's the same process. And we also have the same means of communication by kind of stereotypic action potentials. This is what we do, our own group. It's called the brain scales approach. HBP, the Human Brain Project, appears twice there because both approaches, Spinnaker and brain scales, are pursued in HBP, and I will comment on that. Ours is called a custom mixed signal. Technologically, it's a mixed signal. It's a mixture of digital and analog electronics. Do these things have anything in common? I think yes. They are all massively parallel. That is, they really exploit close to perfect scaling. There is very little a communication overhead. The hardware is optimized for spike-based computing. That's good and bad at the same time. It's optimized because it means if you really want to understand spike-based computing, those systems are wonderful. If you want to do other things, they are useless. So there's a lack of flexibility. I can already say that is the minus sign there. This system has very little, at the end, very little flexibility. They are not general-purpose computers. They're all using asynchronous communication, and we all try to make them as configurable as possible. Even if there's limited flexibility, you still, within that limited flexibility, you want to be very flexible. For example, changing neuron parameters, connectivity, and things like that. In general, I believe that the complementarity of the approaches is very essential. We don't know what the best solution is and which will lead to good results. So I find it very interesting that these things all made it into large-scale systems are now available to the users, so that we have quite interesting times ahead. Let me go through the approaches a little bit more in detail. IBM Almaden, as I said, fully custom, fully digital design. These people really exploit economy of scale by going to 28 nanometer, which amazingly is not IBM anymore, but Samsung, but that's not so interesting here. So they really go to extreme submicron sizes. I mean, there are even better ones now, but 28 nanometers is pretty good. Nobody else is doing that. And they can do it because it's fully digital. They have 100 million neurons on one chip, which is unheard of. It's absolutely spectacular. These are live neurons. Leaky integrated is the most simple neuron model you can think of. It's just integrating information. If you reach a hard threshold, it just fires and resets. Okay? There are 256 million synapses. They are static, however. They cannot implement learning algorithms like STDP, and they are 1 bit. They are on and off. Of course, you can combine them to bigger structures, but a single synapse is 1 bit. They are so-called neurosynaptic cores. That's the architecture. There is 5 billion transistors on a chip, and size-wise is the biggest transistor the biggest chip that IBM has ever produced. They do not have local plasticity, though. Coming to HBP, the Spinnaker approach, it's a many-core system. So as I said, it exploits weak scaling in an extreme way. It's a drastic approach. It has 18 ARM cores on a chip, having, for the time being, only integer arithmetic running at relatively moderate clock speeds. There's a shared system run on the die. On top, as you will see on one of the photos, there is a 128 megabyte DRAM stacked on the die. And the key of the thing, and that should really be pointed out, in order to make this weak scaling approach a success, is the communication infrastructure. So there's a router on the chip, and that router is optimized, highly optimized for one single purpose, to transmit small packets which represent spikes, the timing and the address of spikes. That's really, it's optimized for that kind of computing, and it can transmit 6 million spikes per second per link. Okay, and exploiting this, you can have a real-time simulator, which really where a day of simulation is a day of compute time. And the scaling, because of this optimized communication protocol, really works extremely well. So this is an optimized neuroscience simulation engine, which, as you will see, is very successful. The other approach is the most exotic one, as I said, a physical model. Of course, that's not the circuit we use, but it's the basic idea. You actually use capacitors, resistors, and batteries for the reversal potential. It's continuous time integrating. There is no clock anymore involved. And it's mixed signal analog digital. It's local analog computing and digital or binary continuous time communication. That's, in a way, one neuron, one circuit. So it's a drastic approach to strong scaling. You cannot go any further. But if you work on the single cell execution speed, of course, you can move scaling, the strong scaling barrier forward enormously. And you can do that by choosing parameters. There's a table here comparing biology and VLSI, and by choosing physical parameters, you can accelerate the system. You can impose time constants which are faster than biology. Now, this is just a membrane. Of course, there are other time constants, like fold time and rise time of postsynaptic potentials, plasticity time constants, many others. There are about 20 time constants in the system. If you scale them all by the same factor, you have what we call an accelerated system. Time here is not imposed by an external clock, but really by the selection of the physical parameters. Okay? Just an example here, you see a single neuron bombarded by an input spike train providing excitation and inhibition. And you see here simulation and the response of the hardware. They look kind of similar but not identical. That's very important, of course, not spike by spike. And the second corresponds to 25 microseconds here. So that is what we call the compressed timescale. The HPP is a 30 months project. And there's not much you can do in 30 months. So some people have the impression all this work that I'm going to show now has been done in HPP. That's not true. All projects, actually magically, all projects, True North, Spinnaker, and Brainscale started back in 2005. They didn't know each other at that time, but they all started in 2005. Now we are 10 years later, and we are all more or less in a situation where it starts to be useful. So that's what it takes. To build such a system is not just building a chip. That's easy, not easy, but it's one aspect. You have to make a system, in particular also with all the software tools. What HPP wants to offer is access to these two complementary systems for both, for neuroscience and for machine learning. And there are two machines now, one in Manchester, one in Heidelberg. The Manchester machine at the moment is 500,000 ARM processors with the features I said, and the physical model system at Heidelberg offers individual neurons and synapses 4 million and 1 billion at the accelerated time scale. Let me show you a few photos. Of course, it all starts from chips, and it should remind us that this is not buying things in industry. You really have to start from a single transistor. I mean, in this project, in both, we start from a transistor and we end up with the operating system that we have to provide. So there's a lot of effort. And by now, we also have a lot of unification done, for example, by using the same Pine system, which was mentioned in the introduction. On the left photo, you see a single chip of Spinnaker. Actually, it's not this chip you see there, but the DRAM stacked top, but the 18 cores are underneath. What? No, it's okay. I'm just a bit sick. The physical model machine on the right it looks kind of similar, but it has about the same size. But of course, it's very different conceptually. You see the two white spots there, or gray spots, which are the synaptic fields with about 500K neurons, 100K neurons, 50K neurons each, 100K in total, and about 500 neurons on the chip. Now, the next step is to build systems out of that, large-scale systems to really show, well, we can scale this, not only software-wise, but hardware-wise. This is boards that have been built. On the left side, Spinnaker, 48 chips, about 1,000 cores. On the right side, Brainscales. It's a wafer. We don't cut the wafers into pieces, but we keep the wafers intact under this aluminum frame. There is a 20-centimeter wafers with 200K neurons and 50 million synapses. The next step is to take these boards and scale them up to systems. This is what we have. And on the March 30th, which is just a couple of weeks ago, those systems were put into official operation. They are now part of the human brain HPP platform system. This is the many-core machine in Manchester, 500,000 cores, real-time simulator. On the right, a 20-wafer module in Heidelberg with the parameters I just described. These machines look impressive, but if you want to use them, which is more important, HPP has set up the whole user infrastructure. If you're interested, send a mail, and you can be part of the what's called HPP collaborator. You don't have to be an HPP member. You can just have what's called public access. If you want an HPP identity, which may be useful, you can also do so, and then there are various stages of escalation to get closer into the project. Just send an email and you will get an answer. If you don't like big machines and remote access, there's also another possibility is if you're happy with small systems of the size of a USB plug here, there are two systems now available. For example, the 4 Spinnaker system chips and one of our chips, which is an early version of the HiCAN called Spiky. They sit in little boxes. All you need is a laptop and a USB cable. And we provide you with all the software from configuration, running, and analysis. And then you can run interesting experiments. In fact, most of the papers that have been written with these systems actually come from these little boxes. They are available for sale, for loan. They are also remotely accessible. There are various models here. They're in particular also interesting for students and for universities. You want to train people. Your students, for example, we have complete classes, student classes. If you're a professor at a university, you want to have a little class on neuromorphic computing, we can give you access or even a box like that. If you're interested, contact Steve Ferber or me. Let me talk a little bit about applications now. There's not much time left, but I will be short here. I mean, this is about neural networks, of course. And the typical way of using neural networks is that you have an input pattern, then you have a network state, and you produce an output pattern. And the network state has been acquired by a learning process, mostly supervised learning through some external control, like backpropagation or gradient descent or whatever you have. And in many cases, or in most cases where these things are brought into applications, this network state is deterministic. It's indicated by these two funny dice there. So if you repeat the experiment, it will always be the same, which is important. For example, if you want to apply it for particle physics as a trigger system, which was my past, determinism is the key. You don't want to detect a particle sometimes with some probability. That would be a disaster, okay? You want to be as deterministic as possible. The brain may actually work slightly differently. We know that we can recognize faces and things, but not completely reliably. There is a distribution of patterns which represent reality, I say faces and desks and things like that. And from that, by a stochastic process in learning, but also by a stochastic process in using, both stochastic, I built an internal model of our world, and those are distributions of patterns, distributions of faces and whatever. And so I have what's called prior knowledge, which reflects the reality that I have seen, which is then stored in my brain in probability distributions. I can use those probability distributions in my brain for two purposes. I can use it in a discriminative way. If I see a face, I can say, well, I've seen that guy before, and I know who he is. Okay, that's inference. The other thing is I can even close my eyes. I can completely decouple from the world. There's no sensory input anymore, and I still produce those patterns. You may call that dreaming, for example. Yeah? So I can replay the patterns that I have learned in my life. And I will show you a few examples on how people have done that now with neuromorphic computing. Let's start with deterministic supervised learning, for example. That's sort of the most straightforward way of using neural networks. Now here with spiking neurons. These are not the Kellogg-Pitts neurons or the classical neural networks, but spiking neurons. This is work done by Michael Schmucker with one of our systems, and it's emulating the olfactory system of birds, of insects, of bees. They have sensors for chemical substances which produce sort of a rate-based firing signal depending on the intensity of the chemical substance. Then there are two units. One is a contrast enhancement, bilateral inhibition or decorrelation. There's a circuit for that which has been uncovered by computational neuroscience. Then there is association. You combine these things and say, well, this combination of chemical substances represents a certain flower, and this is done by kind of a winner-take-all circuit, as people call it. So these are the circuit motifs. And the interesting thing is, of course, now that this thing needs to be trained, a learning process, and it's the connection between the decorrelation and the association layer that's being trained through supervised learning. A typical results on a neuromorphic system looks like that. This is a spike pattern plot, where you see time. This is biological time now, by the way, not electronics time, running on the horizontal axis. And there are neuron numbers, like a couple of hundred neurons, and the dots are the firing. And sort of at the bottom, you see these very dense dots. This is the rate coding of the sensor signals. And you see, if you look to the rate, this is the rate histogram there. It doesn't carry any info. It's just a flat distribution, okay? And after training, it's the same flat distribution. And you wouldn't believe that the information is actually hidden in this mess of plots. Now, the activity in the layers between correlation, decorrelation, association is plotted in the middle. And the interesting thing, you see the rate is much, much lower there. It's a very, very sparse rate, almost no activity. And then on the output layer, you get suddenly, again, some kind of a high rate. It carries no information in before training, after training. There are three options the network can distinguish. You see there are two spikes coming up, two rate spikes. Why are there two spikes? Because there's an excitatory and inhibitory population. And you see that the correct decision has been taken typically after, 100 to 200 milliseconds. So the network takes 100 to 200 milliseconds after training to settle to a good solution. This is what people could call multivariate data analysis, which can compete with normal machine learning techniques in a reasonable way. But of course, one may say, well, there is no point. Why would you do that? Why would you do it with spike? Well, actually, as I will say later, of course, there is the energy advantage and there is the timing advantage. I will comment on that later. And what we see here is that it actually works. There is another example. This is a bird. It's an owl. It can locate sound sources by measuring phase differences between the left and the right ear. This is an example for deterministic unsupervised learning. The idea has been published a long time ago by Konichi and others. And what we see here, how it works, basically there's a flight time to the right and the left ear, and on the right picture here, the flight time to the right ear is short and to the left ear is long because the mouse or whatever is on the right side of the body. And then by a correlation method, coincidence method, in the brain, these are neurons there, this flight time is compensated by finding the coincidence by long flight time from the right ear and the short one from the left ear. There's a circuit that can be implemented on a neuromorphic chip. Without going into details here, it's really implementing this coincidence by what's called phase locking by spike timing dependent plasticity. As a side remark, this is using analog synapses, which are pretty bad, actually. These are bad analog circuits. Okay, this is a variability of a typical parameter, this histogram there, which you see is around 20% or so if you use many synapses. The interesting thing is that the learning process, which is unsupervised, this is the development of the synaptic weights there, it works and delivers a phase detection with a precision of about a couple of nanoseconds with really bad analog circuits by really self-calibrating the system by a plasticity mechanism taken from nature. And that even has some technical applications, like, for example, you can apply that to ultrasound emitters and sensors and use it for echolocation, and we are working on that now. So this is a small system, of course, but it's an example for unsupervised learning. There is the other thing, of course, the stochasticity. This is the last animal I'm going to show you. And what you see there is this famous picture, and it shows that what you do when you recognize things, when you infer, what you see is that you use pre-existing knowledge that you have acquired during your life about animals, but you see that you're not so sure here, it could be a duck or a rabbit, and you never see both at the same time. You constantly take samples from something that's stored in your brain, and you come to a conclusion that it's a brain or a rabbit. Now, one way to implement this or similar approaches is through so-called Boltzmann machines, which are a network of symmetrically connected stochastic neurons. Stochasticity, of course, is the question, where does it come from? Is it the cells or is it the network? We believe, or one of our implementation is that the noise is actually coming through the network itself. I can answer questions, but will not further describe that here. Those are binary random, neurons represent, or nodes represent binary random variables, which is also not so clear. How can a spiking neuron be a binary random variable? Well, it can, for example, if you represent the two states by being active or being in the refractory state, then the probability for a state vector converges to a target Boltzmann distribution, provided that you have a given temperature, temperature being the noise in the system, okay? And of course, there's an energy function, which for the physicists, this is pretty much what you see in the Ising model, where the energy function is very much determined by the weights between nodes, and this is what you, of course, train then in your supervised training mechanism. What is it for? Well, it's to learn internal stochastic model of your input space, and then let it be generative or inferring things. So there are two steps you have to go through, which is learning and using. Learning is slow, whatever you do, and using is fast. How do you learn? You clamp visible input units to the value of a particular pattern. You reach thermal equilibrium. This is what the Boltzmann machine does. You increment the interaction between any two nodes that are both on, and then you keep repeating this procedure for a while. Then once you are done, this is a long process. It can be, of course, fast with neuromorphic systems. Once you are done, you run the network freely and sample from those probability distributions. There are two ways to do it. On the left side, you see an example doing this in a generative mode, a free-running or dreaming machine without input, just recapitulating what it has seen, and you see it spends most of its time in this space of three numbers at the four numbers that it has seen, and then thinks that it sees all these things there. On the right thing, you have a discriminative mode where we provide an input patterns which are only compatible with 4s and 3s and not with 0, and then the machine actually mostly spends time in the 4 and the 3 location. So this is very interesting. Does it also work in hardware? Yes, actually it does. This is a hardware experiment where you see the red thing is a learned distribution. This is after the learning we are now doing. The application, we are sampling from the system, and you see in a relatively short time the sample distribution approaches the learned distribution. That is, if you look at things again and again and again, you suddenly recognize and you make a decision, for example. The interesting thing is that this is really fast, although it looks slow here. This is the Kullback-Leibniz divergence between the learned and the sample distribution. It's a log scale. In the real experiment on hardware, it's on a millisecond scale. So there's a huge acceleration with respect to natural time scales. This is now being applied to, I'm almost done, yeah. This is now being applied to really more complex neural networks, for example, having the full set of MNIST handwritten data sets. And there is an interesting observation, which has now been published very recently by those two authors here, is to use these Boltzmann machines in not quite deep learning, but structured networks with a hierarchical thing where you have a visible input layer, a hidden layer that codes the receptive fields of the hidden layer you see there in the middle, and the receptive fields of so-called label units, which then decide whether the output is any given number. And so it works very well. As you can see, the label layer is up there where you switch between 3 and 2 of some handwritten characters. The interesting thing that people have observed, and this is a very new observation, is that in learning, which is going through the energy landscape of the Boltzmann machine, often you get stuck in this energy landscape. And there are various methods to overcome that, like simulated annealing, many machine learning methods. They are all computationally very expensive. It takes a lot of time to apply them, and it takes a lot of energy to pay for them. What we have seen now is that biological principles, which people so far didn't really know what they are good for, like short-term plasticity, for example, or the high conductance state of spiking neural networks, actually improves the mixing. That means going over a barrier in a multimodal energy landscape. And that's very interesting because that may be the final answer why spikes are important for computing. Of course, there is the energy argument, which is always there. If you have sparse coding, use less energy, that's all very interesting. But there may be a computational reason. It really accelerates learning. And to me, that is a very fundamental thing, because if you look to applications of deep learning that people do now, playing Go or whatever, it's all great. But what people don't see is that it's a long, long, long period of very expensive use of piles of graphics cards running for months at 100 kilowatt level or even more, and you pay a lot of energy and time. It's not very practical. And here, for the first time, I think we see a way that biological principles can really be transferred into machine learning and make computing a lot faster. Let me come to the end. I showed you this plot before, energy used for synaptic transmission. This is a log scale. The neuromorphic systems kind of fill the gap between conventional computing and biological computing. You see Spinnaker here, 10 to the minus 8, Joule, the brain scale 10 to the minus 10. This is far, far away from both, from the brain and from the conventional computers, but it's a very convenient gap because it's very interesting. It really makes these things worthwhile to construct also on a larger scale. There's a huge amount of energy to save. Timescales, I said that to me are the most fundamental thing. There are many timescales in nature. I have 12 orders of magnitude here. Causality detection in synapses at the 100 microsecond level. That's also the steps you do in your simulations, for example. Developmental timescales may reach years, okay? In simulation, of course, thousands of seconds are easily accessible, no problem, with a slowdown factor of 1,000. 1,000 years are definitely not. It's just fundamentally excluded, okay? Whereas accelerated, or at least real-time models, really provide access to different timescales, which to me is the key. I don't want to talk about evolution. There are roadmaps in HBP to improve the systems. This is only the first approach. We have the time until 2023 ahead of us. This is the Spinnaker roadmap leading to a chip called Spinnaker 2, which is basically a factor 10 better in all the parameters at fixed energy cost. Okay, there will be floating point operations, many other aspects, the new memory, HMC memory interface, and things like that, which I can answer questions, but I have no time to discuss. On the brain scale system, we will also introduce 2 new features, which are probably better seen on this slide. First of all, and most importantly, going digital to some extent, or going flexible to some extent. Each of the little chips on the wafer will have a PowerPC processor now. There will be 400 such processors on the wafer. We go to 65 nanometer process technology. Those processors will provide completely flexible local learning rules. So you can do old STDP, of course, but you can also do rewiring on the fly. You can have any model for neuronal or synaptic plasticity with slow and fast circuit dynamics. We are also working on introducing active neurons, structured neurons with active dendrites, with a spatial structure, to use neurons as complex pattern detectors. For example, the HTM model, hierarchical temporal memory model for prediction making, currently under development by Numenta company in the group of Jeff Hawkins in California. We are working with them on that implementation. Yesterday we heard a lot about technology. I like technology a lot. I will close with a technology slide. If you have seen our systems, they are kind of still very conventional in terms of electronics manufacturing technology. We are now working with Fraunhofer to, for example, embed wafers and also chips into printed circuit boards so that large-scale production can be much more efficient and much cheaper and much less time to assemble these systems. This is a really cool technology. I'm just showing it to you. My conclusions is after 10 years of development, it took a while, we now have hardware systems that are available that have reached a high degree of maturity, ready for non-expert use cases. They have a very high degree of configurability, which is important, with dedicated software tools, but obviously they are not and will never be a replacement for general purpose computing. They may be useful for neuroscience and machine learning, but there are other things that you will certainly never do, like, for example, simulating materials or weather prediction. The only way to access multiple timescales in brain circuits, which are present in those systems, is using these kind of approaches. And I think that's the only way to make them functional. This is why I think it's so important. They are well suited for stochastic inference computing, and they're also well suited for use of deep submicron and non-CMOS devices because of the fault tolerance, which I didn't demonstrate here, but which is definitely there, and we have evidence for that. Thank you very much.